Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bitesize Bio.
Welcome to the very first episode of Flow Stars, where we interview top names in flow cytometry to get to know them on a more personal level. This first interview is a double headliner with Tim Bushnell over from Rochester Medical Centre and Alfonso Blanco from University College Dublin. In this episode, we discover the importance of training in a core facility.
If you get people to understand how to use a machine, they don't break them.
Tips for writing up your thesis.
I got out of the lab. I literally moved away from the lab so I could focus on the writing.
And the real reason Alfonso wanted to do a PhD in botany.
Because that will allow me to go as well outside the lab. To go outside, climbing mountains or walking around and trying to pick up the plants for your analysis.
Hi, I'm Peter O'Toole, and today I'm joined by Tim Bushnell and from Alfonso Blanco. Thank you. Hello, both.
Hey.
Hi, Peter.
Different time zones, with Alfonso over in the Republic of Ireland, Tim over in the USA. So this is a bit confusing time. I guess one of you is in a completely different time point of day. So which one of you is drinking wine? Which one of you is having a cup of tea?
I'll drink wine.
You take my wine.
So, guys, you know each other really well. Where did you first meet?
I was over in Dublin to give a training course with a company called FlowSight, and it had been sponsored by Accuray. So that was the first time that the two of us met, and I think we hit it off then. And ever since then, I've been causing antics around the world as much as we can.
And more.
So it was a memorable meet then.
Very true.
Yeah. So you must have been to Ireland more than once, Tim?
I think I've been to Ireland now, 3 times, Alfonso?
Yep. And we tried to bring you a couple of times more, but not successfully.
So
stop by.
Does this explain the what is this exactly?
On my time, I went over to the Guinness Brewing store. I was there to find William Gossett's original journals to study about how he developed the t-test. But while I was there, I had a couple of beers.
So you got a master's brewer license at that point.
they give them out to everybody. So it's.
And you didn't have to sort their yeast into different profiles so they could get different Guinnesses and different strains.
They weren't too receptive to that.
Yeah, that would have been quite good, I think. I know. Cell sorting yeast is fairly, maybe not a thing that everyone wants to do on their own sorter. So you've done a lot of work together since, I believe.
Yeah, that was the first time that Tim came here was for a training course that we organized. And then I was telling him I really like training. And then he recruited me as a flow site instructor. And we organized a training course in Lisbon with Rui Garner. And then we went together over there. He was my main instructor. He was basically checking the list. Alfonso can teach. Yeah, he knows how to teach. No clue. So he was checking putting the tick boxes, but it was a good time together. And since then, we have been working together all the time, and just running, organizing courses, making mess and things like that. So that's
actually, I'm just thinking, how did you learn? Where were your skills picked up from of how to become a good trainer, Alfonso?
I'm a good trainer.
I think the first time we met might have been at York.
Yeah, that's true. But that was not a casualty, to be honest, Peter. I was looking around because I like training. So I was looking around where is the best training courses. So I realized that yours were super popular. So I went to your site to check. And I told you on the first day, I was honest with you. I want to go there to learn how you teach, to copy you. And that's exactly what I did. I just copy you, your scheme. I not just that, also. And this is funny when we are talking about trainings. Every time that you train, even if it's the same material, you always learn from the others. It doesn't matter, it's the same slide. Somebody comes with some different point of view, with a different approach, with a different example, different way of explaining. And it's what I really love of training with other people. So I really enjoy your training, you know.
Yeah, it's unique, maybe. Or maybe not so unique if you've copied me.
No, I copied and I made my version. Less jokes per minute.
And what about yourself, Tim? Because obviously you do a huge amount of teaching as well.
I probably have to credit back to the fact that I like to play in front of an audience. In high school, I was involved in debate, Model United Nations. Did that in college. As a grad student, I had to teach for my supper, as it were, and it just is natural. And when I took over my first core facility, it became apparent that the training was the most important, because if you get people to understand how to use a machine, they don't break them. And that's a huge lifesaver. And it's just, continuing to learn, as Alfonso said, learning from other people, learning from, the community. We've got Flow's got a great community of teachers and educators. We all want to, and we all do. And, we put our different spins on it, but at the end of the day, we say the same thing. It's just a matter of the flamboyance of it, I think.
Yeah, so that's an interesting point. You mentioned the flamboyance of it. You give the same talk. If you're teaching, I know this, you give the same basics, at least, talk every time. And that's a lot of time given exactly the same script. I feel it's a bit like being in a theatre. You have to do the same performance every time with the same enthusiasm. And actually, to be fair, maybe it's not that hard, because you look at the delegates and they're enthusiastic. So I become enthusiastic.
Absolutely.
But you do have to be a performer to really get the engagement. Which comes really actually to some pictures that Alfonso sent to me. So tell us about this, Alfonso.
That's a really funny story, because I'm born in Seville. That's the south of Spain. That's where all the funny people is in Spain. And the accent is just for comedians, to be honest. They speak with the set. And that's because my parents, they were working and they were living there for many years. But my family is from the north side, from Galicia. So we moved when I was 7 back to Vigo, my hometown. And in fact, I consider myself from Galicia. And the accent is completely different. It's always, they go like that, talking like that, kind of Irish. So, in fact, so when I arrived to school, it was not an easy time. Also because the color of my skin, I'm really dark. And in the south, it was a lot of sun. I arrived there with the dark skin, and everyone was looking at me with my surname Blanco, which means white. And it's not matching the white. So the nickname, it's clear. I was negro all the time. So that was me. That was my starting point. But I'm not a good flamenco dancer. No. Don't go that way.
So I know you very well from the dance floors at Cyto. I don't think I've got a picture of us, but I know you've got a picture of us.
I have a picture of us.
But come on. Your dancing obviously went up a step. And this is, what? Did you explain?
That's in Santiago de Compostela. That's where I was studying. And this is a tradition. And in fact, it was happening 2 days ago. Basically, you put a big bonfire, and it's good for cleaning up your bad spirits around you to jump over them. And you have to jump at least 3 times. Of course, I was jumping a lot. You can see my runners, and that's because I was playing handball. So those are the handball runners.
How long ago was this?
This was a lot. I was 22 years old at that time.
And you're still wearing the same shirt?
I'm still wearing the same one. I haven't grown up that much.
So talking of dancing, Tim, this is a picture of you up a pole. So is this your equivalent of pole dancing?
It was actually, I was a Boy Scout and took a trip to camp in Philmont, somewhere in New Mexico. And one of the challenges, I had different camps there, and one was a logging camp. And that was learning how to climb a pole, doing spur climbing, which is just with a couple of spikes and a belt, and kind of a little bit of conquering a fear going up a 50-foot pole by your, to get to the top. And haven't done it since, but did conquer it back then.
Yeah, I don't envy that at all. And this is your scout group, is it?
Yeah, this is the group that I went with. You'll recognize me because I don't have a beard at the time.
That doesn't help.
Yeah, that was a great trip. I had earned my Eagle Scout award, the highest award in the U.S., a couple of like 6 months before I went on that trip.
Getting back onto work, a little bit. So I'll start with yourself, Tim. What have you found the most challenging time in your career?
Other than right now with COVID, I think for me, the transitions from college to grad school and then finishing up my PhD were two of the more challenging times. In grad school, I took on a lot of projects trying to figure out what I wanted to do, but eventually had to coalesce to one, and came to a point where I needed to finish. And so it was a lot of work going on, but I had a newborn baby and everything. So it was that was a very challenging time, both scientifically and personally.
And so when it came to finishing off your PhD, which I know some people watching today will be in a similar position or not, well, how did you cope with that? What mechanisms did you put into place? What tips would you give to people in that position today? The biggest one is I got out of the lab. I literally moved away from the lab so I could focus on the writing. And I wasn't, and that took a lot of effort because as a scientist, you want to be in the lab. And, I'm a kind of guy who wants to be sitting there figuring out what the next thing is and going on in that direction. So you have to put a stop at it, and separating yourself physically made that possible. Yeah, I did a similar thing. I actually had to buy a PC when PCs were not something everyone had at the time. And yeah, it made a big difference working from home at that point, which during lockdown, gosh, what we all do now, isn't it? Well, some of us work at home, other of us working, I know, somewhere out in space, looking at Tim's background.
They talk about social isolation. I think I'm pretty isolated right now.
Certainly out of this world, Tim. Alfonso, what about yourself? Most challenging time?
Clearly it was a few years ago. And we were talking before about teaching and training people. And you want to have on the other side people that really want to learn. And it's quite hard when you have to deal with someone that doesn't want to learn.
And you just want to help them as much as possible, but sometimes you don't get there, I guess.
No, and if you don't want to get there, that's even worse. And when you don't want to be guided into the right direction, it's even worse. And they don't recognize those situations, that's even worse. So the first thing is recognizing. And usually we know that we cannot know everything. It's impossible that we can know everything. But that's the first thing, the first step of recognizing. When you think you're too clever for everything, that's a bad thing.
I've been really lucky. York has been exceptional. I've not had those sort of problems to have to deal with firsthand, which is, fortunate. What about yourself, Tim? Have you encountered similar?
Personal, becoming a manager is a different skill set than being a bench scientist. And it's funny, as a PhD, you're trained in being a scientist and working at the bench and developing a hypothesis and all that. As you move in your career, you're going to, you become a manager. And so now you have to manage people. You have to manage their expectations. You have to manage their education. You have to motivate them. The hardest thing I think is actually the interview, finding the right person. I've always, in the staff that we've hired, we've not looked for somebody who has flow experience because that's what we can teach. It's finding those intangibles, customer service, enthusiasm, and bringing them on board, and then supporting them, and always being supportive of their goals and checking in and saying, what do you want to do? Is this going to be your career, or is this a stepping stone? And supporting that growth is, I think, the hallmark of a good manager. And when you have a situation like Alfonso, and I've had similar situations too, it just, it devastates you because of the effort that you've put in, and it devastates you because you question, am I good at this? Am I doing the right thing? So.
Yeah. So actually, I'm just thinking about Alfonso, and you sent me this picture, and obviously a very keen team player. And it is important to find the right, gut instinct is sometimes the best thing to go on. Yep. If they've got the personality, they can often carry around the role, hopefully, but not all. Maybe. So Alfonso, what's this picture of?
So that was my last team, and that was my last game playing handball, which is kind of indoor soccer. You play with the hands and you kick everyone, which is quite funny because that's really a release of stress. In the picture, I'm in the corner, but I was in the middle in the defense. I was really bad trying to score, but I was really good defending. and I was kind of controlling everyone, putting them into the right position. And that was my last team because after the picture, we started to play, and I have a really bad injury in the knee. I broke the triad, so the 3 ligaments were absolutely broken. So that was my end of the career of handball, but that was really successful story. Lots of friends. We're still meeting each other every time that we have an opportunity. We have a barbecue, lots of beers, sharing stories like old guys. Oh, you remember when you kicked this guy? Oh yeah, he end up in the hospital and things like that. So that's good. So
there's a lot of mention of kicking.
Yeah.
and where did this come from then, Alfonso? It's just this innate ability to kick people out of your lab, like you were just saying about, or
look at my hands. So when I'm saying to my users, "Don't do that, or I will kill you," it's real.
Well, do you have this poster around on your labs, on your flow labs? Say, look.
So that was before handball. I was practicing football. I was not good at football, and then I moved into taekwondo. That was a new sport in my school, and I was really good in taekwondo. I arrived to the green belt, and I was getting ready for fights for the Olympics, Barcelona '92, and I have another injury in the knee. They told me that I have to stop practicing taekwondo, and at the end was a wrong decision because I should keep going and reinforcing even more the leg. But that was too late for coming back.
I've noticed you get very good at sports that not very many people play.
Sensational.
So seriously, I know there's some highlights. I asked you the difficult question about the most challenging time. What's been some of the highlights of your careers? This time, back to Tim on this one. What are your highlights of your career?
I think I have to say, the biggest highlight was when the transition from postdoc to faculty, where I became the director of a core facility. I was hired by this recruit at the university who was starting a small core at the time, and the job was support the 5 or 6 labs that were there, and then the rest of your time was free. And so I had plenty of time to really play and get into the guts of the technology and get into the guts of what was going on, and just really enjoy this opportunity to work with people and get them to a point where that they could then go off and finish it. So kind of that first explorer discovery, and then, okay, here, you go finish the work now. Here's the protocol. So that was probably, I think, my favorite, 5-6 years.
Yeah.
How long? Yeah, about 5 years I was in that position.
That's and what was your first cytometer then?
As a postdoc, I before becoming a postdoc, I was a plant biochemist, molecular biologist. So I had heard of flow cytometers because a colleague of mine was a marine biologist, and they took one on a ship, but we never had one. I moved into B cell development because there were no jobs for plant biologists at the time, and worked on a 3-color effects caliber. And it was like this, it was my Star Trek tricorders, I always tell people. I put cells in, I got these beautiful answers out, and for the 5 years of the postdoc, I did everything by flow cytometry.
Yeah, but that insinuates it was all just science fiction coming out of it.
It seems like that at times, but there's a little bit of science fact in there too.
Yeah, my first was a caliber as well. 4-color. Oh, get us.
When we got our 4-color, when we got the second laser, I was over the moon.
Yeah.
Look what I can do.
Yeah, that was a beauty. Always fond of the first system. What about yourself, Alfonso? Start with, what was your first cytometer?
My first cytometer was a big monster. It was a Culture Epics Elite, and it was a sorter. And the chair was super comfortable for Spanish, to be honest. It was in a botany department, and I just heard about it because when I was doing my degree, on the last year, I had to do a job for getting points and things like that. It was polyploidy in plants. And the teacher told me that the university has bought that machine that has lasers and computers. And I was like, oh, wow, cool. I want to go there. I was not botanist, and I was not really into the plants, but just the lasers and the computers called my attention. So when I arrived and I saw all these things and touchable screen, I just fell in love with that. And that was my first instrument.
And what's been your favorite instrument over all your career?
My favorite one was the cyan, to be honest. And there were quite a few things that I really love on the cyan. And I still, in fact, I still have two of them here.
I've still got two.
And they're fantastic. At the time, were so robust and so fast, so easy to use. The software, I love the software. And the fact is, I was doing a lot of teaching and training with the software, even with data from another computers. I just love science.
Yeah, I think the MoFlow legacy has a lot to go with.
Mm-hmm.
I think it would scare most people now. That was, you were just still one with a legacy. You just felt it, breathed it. It was just an extension of you. Literally, it was like a growth to you on your hands. I kind of missed that. But yeah, I haven't lost any cyans yet, so I haven't had to say goodbye to them.
No.
But yeah, the legacy was sad to see that one go for us. So what's been the highlight of your career, Alfonso? I've heard about your low time, and that I heard that just how stressful that was. You could feel it in how you were expressing it. What about the highlights?
Highlights, too many.
I've got some. Look in the side of my head. Oh no, actually no, just great. It's not really the highlights.
So there are quite a few things. If I have to pick up one, is quite difficult. But I would say one is a share highlight with Barry Moran. We organized here the ESCA meeting back in 2011, and it was a very successful meeting. We made lots of friends. Was quite risky because we hired a center. The conference center of Dublin was still under construction when we decided to go for it, and the construction is not the best thing sometimes in Ireland, but we met the vets, and we were quite successful there, and it was really a big effort between Barry and I to put together that. But also at that time, there was something that we tried to put together, and it was quite challenging. And it was the first sorting course with 14 different instruments on site without being on site. And now it's back into place because we were doing that sorting course remotely. So that was with John Tiggs, Basilis, Rick Garner, John Daly, Barry Moran, and myself, and was with Mark Cheaton as well. And we put that course together, and we connected 14 different sites to train 32 people in a very small room. That was, again, a big challenge. And when we finished, and not even when we finished, when we ran the course, it was so fantastic, the feeling. So it was definitely one of the best situations I ever been.
So Works obviously has its highs, it has its lows. Usually long hours, I've got to say. I think we both appreciate that one. What do you do outside of work to take your mind off work? What's your other focus? So Alfonso, I'll start with you on this one.
First, family. I enjoy a lot of the kids, playing around with them. The other thing that I really love is traveling. I entertain traveling, looking at sites, walking around places, and I don't care if it's a city or if it's nature. And if I can do it with the family, it's even better. If not, I go alone. There you go. That was one of the reasons why I wanted to do PhD in botany, because that will allow me to go as well outside the lab, to go outside climbing mountains or walking around and trying to pick up the plants for your analysis. That was also quite funny. So that was really the complement for my job. I wanted to be in the lab. I wanted to be in the nature. I wanted to walk around. I don't want to be supervised all the time and stuck into some timing. So I found the perfect balance in doing these things. And this was in the north of Spain, in Somiedo, where I have to pick up those plants, my ferns, isolate each one of them and classify them. So we use them. If you see them, they're ferns. There are not many characteristics around them, but they have different DNA content. So they go for 222 pairs of chromosomes. So counting them was difficult, and that's why flow cytometry ended up. So I was doing flow cytometry of each one of them to quantify the amount of DNA on the plants. And that was my beginning of cytometry, with those little ones, I have to water every single day, make sure that they don't have any worms eating them. And it was quite funny with my father, because I told him that I have to go to the greenhouse. When he brought me to the greenhouse, he thought he loved flowers. He thought that it would be full of flowers, and we have ferns and grass. So he's like, what is that? This is a greenhouse. Yeah, that's what I'm working. You will not end up anywhere with this.
Like I said, I think the picture is just fairly typical. It feels sometimes when I go to conferences, I just have Alfonso on my shoulder constantly talking to me.
Correct.
It's mostly family. Any other activities out of work?
No, not really.
Yeah, I could get that. And Tim, what about yourself? I think, were you a farmer once?
I'm a farmer wannabe. That was one of my first houses. We had about half an acre of property, and I've got an acre now. And I'm not the best green thumb, even though I grew plants. And my plants were water ferns, so I didn't have to water them like Alfonso did. But this is funny because I don't even remember why I shaved off my beard. And my kids, when they saw me for the first time, didn't know who I was. They were scared. So never again.
Actually, I remember my dad always had a beard, and he shaved that off. It was very weird, very strange. Yeah, you don't know. As a child, it confuses you a lot. But yeah, parents, why do they do things like that to us? I have no idea. So, what else, Tim? What else outside of work do you do?
Like Alfonso, I like to travel. We try to do a lot of stuff locally. I like to cook. I like to barbecue. I like to teach, be with my kids and teach them. Still active, involved in the Boy Scouts with my son's troop. And I've been a brewer on and off for about 20 years as well. When the mood strikes me, I haven't, I've done meads, I've done ciders. I'm in apple country in New York, so it's always great to get fresh pressed apples and make cider and, of course, beer. My favorite times is we'd homeschooled our children in the U.S. And as a science project, I taught my son how to brew because it was aseptic technique, it was microbiology, and it was great because I had to drink the results for him. Oh, darn.
So this is your son, is that correct?
That's my youngest son. That's Nathan. Took him to England last year for the first time, visit a friend of mine. We just got off the red eye in the taxi getting to the hotel, and shortly thereafter we were downtown London, hitting the eye, going over to the various sites for a couple of days, including they have a Jack the Ripper tour, which was interesting to walk the streets where those crimes were committed. But it was extremely hot too. It was like the heat wave last year that you had. So it was 95 degrees, and you're walking through, a city which is hot anyway because of all the pavement.
What time of year was that?
It was end of August last year.
Okay. Yeah, last summer was our hottest for a long time.
Yeah, we eventually made it down to the Bournemouth area, and my son's into military, so we went to the tank museum down there, and he had an absolute blast.
I'm trying to think what the tank museum's called down there. Can you remember what it's called?
I was the tank museum. It's in the.
I need my son to come in and tell me. Yeah, we've been there. It's a museum as well.
Yeah, they had, he was excited to be able to see, the first tanks and stuff. And this year in his schooling, they studied World War I, World War II, and he just, was really had a lot of additional facts to provide to the class and stuff. So,
yeah, it's amazing. It's full of tanks.
Yes.
When you go as well, including the demonstrations. I actually have them running as well.
Very true. Yeah.
Just going to, again, back to the day job for a minute. We've heard about challenging times. We've heard how you got into flow cytometry itself. How did you find core management? Because how long have you both been running a core facility? So, Alfonso, how long have you been running a core?
It was 16 years, 1 month, 3 days. So,
okay. I'm still counting down to retire. Is that a countdown to retirement or just a day up from the day?
No, I have to say it's a hobby more than a job. And that's what makes it nicer for me. It was quite funny because I was doing my PhD. It was a long PhD. It was almost 9 years in PhD and master degree. And at the end of that period of time, I was in France, in Paris. And I had a supervisor there in Paris 6, Sonia Silvia Yakovlev. And after 3 months there, she told me, look, Alfonso, can I talk to you? I said, yeah, sure. Look, you are not a good scientist. What? You will never be a good scientist. You're not, you cannot be a researcher. I said, 9 years of PhD. Come on, what are you talking? Oh, you're a good technician. I say, no, I'm not a technician. I cannot be a technician. No, but you know, because I looking at you, you're going all around, looking at everyone, what they're doing, how they're doing. You're helping them with imaging, chopping things. And you want to learn how to do it and then improve them. And these are a kind of job that you could be good at. I'm like, yeah, but still a technician. No, but they have good jobs. They have permanence. They are well paid. I say, oh, wow, now we are talking. Now we are talking. As Tim said, after that period of time when you're writing the thesis, and I was isolated as well and writing my PhD, I was thinking on what he said. I said, at the end of the day, I want a job. Science is good, but I want a job. And I applied for a job. And I remember my first position, kind of a core manager. I applied for that one in Southampton in marine biology. And I went to the interview. It was the worst interview ever. If you think on the movie, what's the name? Billy Elliot, the guy that wants to be a dancer. I want to be a dancer, a ballet dancer, but with flow cytometry. They want to put someone in a submarine, shipping them all around the world, every single ocean. That's exactly what I want. This kind of matching what I told you before, right? So that was a dream. And that dream didn't come through because I couldn't dance. So I was just absolutely blocked. As soon as they said, you have 30 minutes for our question, turn on the timer, I couldn't even talk. I was just blocked, absolutely blocked.
Wow.
Second interview was here in Dublin, changed completely the style. I was far more relaxed, and I started here.
I'm so gobsmacked that something actually made you quiet, Alfonso. Tim will back me up here. You have a reputation of never stopping.
Very, very true.
That I've got to say, whoever it was who interviewed you, I want to know who they are and find out exactly what they said to stop you talking.
They stopped me talking. It was quite intimidating. You have the light here, dark room, 3 guys on the back. I'm professor whatever. And you have 30 minutes. This is the job description, blah, blah, blah. And you have 30 minutes for questioning. And we will ask you 30 minutes, and the time starts now. Suddenly it was like, everything was just erased from my mind. I have no idea how they did it.
Wow.
They stopped.
Brilliant. I'm full of admiration. Tim, what about you? How long have you been in the core business?
I've been in the core since 2003, so 17 years. Again, at the end of my postdoc, I didn't know what I wanted to do with my life. I knew I wasn't going to be a traditional scientist, much like Alfonso. I'm like discovering things. I like improving things, but as we preach about all these additional experiments and confirmation, I'm not as good at that. I like to explore. And so I started exploring the options of joining the core facility when this guy got hired at university and hired me and basically turned me loose. From that, in 2008, I took over the course here, the flow core at the university. And in 2012, since 2012, I've been overseeing all of the major facilities here at the university. So I have 7 core facilities that report to me.
Yeah. It's similar here. But so what are those core facilities? Give them a shout out.
So I've got a genomics facility. We've got a confocal facility. We've got a multiphoton facility, an EM facility, and a proteomics. Who else am I missing? Flow, obviously. Can't forget Flow. And we have recently transitioned our what we'll call our mouse genome editing. Our transgenic core is undergoing some transition at the moment. But it's going up that ladder of finding how to administrate and be a businessman. You know, in an organization where you're a nonprofit and having all those pressures. I mean, I'm fortunate. Growing up, my father worked for Xerox and was a senior executive there. So back in the 80s, I was reading all the books he was. You know, everything from the Book of the Five Rings and The Art of War, which I still haven't figured out how that applies to business, but that was the big thing in the 80s in U.S. Business culture.
Actually, that brings me to some quick fire questions, if you don't mind.
Fair enough.
Okay. So, Tim, I'm going to start with you on this. Okay. So very quick answers. Tidy or messy?
Messy.
Tea or coffee?
Yes.
Beer or wine?
Beer.
Yeah, you can't say yes on that one. Sweet or savory?
Savory. But I want to.
Well, that's obvious because obviously in America you don't really do great chocolate, do you? But
again, it depends. I like both.
okay, well, you got to choose one.
Okay, if we got to choose one book.
Okay, what are you reading at the moment? No, it's not quick fire.
No, right now I'm reading a book about negotiations, or rereading a book about negotiations. Getting
factual books rather than fiction.
Mixture of both. A lot of science fiction. A lot of you know. I went through a Tom Clancy phase recently. I rereading some of Tom Clancy's books, but I just finished an MBA, so I spent a lot. 2.5 years reading nothing but business books, and going back to refining and improving my leadership skills, which have been invaluable in this whole COVID crisis.
Okay. So actually those books do sound a bit like a mix of fact and fiction themselves.
That's very true. They are.
Alfonso, are you ready?
Ready.
Yeah, some quick fires. Eat in, eat out.
Eat in.
Cook or wash up?
Cook.
Is that because your wife can't cook?
My wife cooks really well, so she doesn't allow me to cook, so I have to wash up.
I was just trying to get you into trouble. You a beach bum or do you prefer sightseeing?
Say again?
Beach bum or sightseeing when you go on holiday?
Sightseeing.
Sorry?
Sightseeing.
Sightseeing.
Yeah.
Home or being at work?
Home.
That's a lucky answer as well. You'd have got in trouble for that. Yeah. Ireland or Spain?
Spain.
Oh! UCD are going to fire you tomorrow, Alfonso.
I know. Spain is Spain. It's in the blood. I cannot deny it. And in fact, one of the things we are running here is the Spanish Research Society in Ireland. So it's trying to create links between Ireland and Spain.
Yeah.
It's trying to make a bridge in many different aspects. And one of them, for instance, is trying to learn what Ireland has really good and trying to bring that into Spain. So we are making some kind of connections with the government. They're asking us for our feedback around research, but also we are trying to bring things that we have here, we have in Spain back into Ireland. And one of them is, of course, education and bringing new students in, et cetera, et cetera. So I'm divided. In fact, you can see quite a few of my pictures with a double flag, Ireland and Spain. And it's funny because sometimes I speak, we, the Irish, are doing this thing. And on the other side, I'm saying I'm Spanish. So after 16 years, I feel quite Irish. I always felt quite Irish.
It's quite a big cultural change coming over to Ireland. How did you find that?
It's not difficult because the Irish are quite Celtic, and Galicia is Celtic. And in fact, we are the Celts. The Celts were the first inhabitants here in Ireland. So the way of doing things of the Irish is quite Galician. I mentioned before, the way we talk in Galicia, talking like that is kind of Irish. So from that point of view, even when you ask anyone in Spain how a Galician person answer a question, they never answer. They answer the question with a question. So if you say, are you going up or down? And you, are you coming up or down? So that's the same here. How are you? What about you? So it's not difficult to adapt. There are differences. Of course, there are differences. Well, I feel half and half.
So any advice? Because obviously when you come out to a PhD, one of the big questions you have is, shall I go abroad and do my postdoc, or shall I stay in the UK? Yeah, stay in the home country.
Yeah.
And there's a lot of pressure, I think. Going overseas really boosts your CV if you're successful when you come back. Obviously you don't have to. I certainly didn't. I had the offer, and then the York job came up, and that was more attractive.
Yeah.
So were you not afraid or scared or worried or?
I was, of course I was. I remember there was not so much internet, so I have no clue how Ireland looked like. I arrived to the interview, get the bus to bring me to UCD and take the interview. They gave me the position on the same day, which is kind of shocking. I thought that I was forgetting something. Could you come back? What I've lost. And then I came back, and the only thing they gave me was a list of telephone numbers and for finding a location. So I arrived to the airport with the luggage and a list of telephone numbers for where to stay. So it was kind of scary because I was not quite sure what I will face. Nowadays, I will really encourage everyone to go. It's quite simple, and especially now with the Spanish societies like we have 18 different societies, internet, Facebook, everyone is helping everyone to move, and they are telling you all the tricks and tips to move, accommodate, how you have to do with the administration, and that's something that I was really missing when I moved here. So now societies, embassies. Now I'm working and dealing with them. They're extremely helpful. You think, okay, I'll go there when I lose my passport. To be honest, there's a lot of work behind the embassies. And I really recommend people, if they're moving away or they're going abroad, the first person to talk is the embassy. And they will provide you huge amount of information. Really.
I think just moving aside from that a little bit, part of that success is the fact that you found and developed a network to support.
Absolutely.
And I think in our careers, whether it's academic or going into the core side of the business, or the shared resource type side of business, networks are vital if you really want to succeed, I think. I think they're very influential. And turning over to Tim now, actually, you have an online resource.
Yeah, that's true.
Hugely useful for looking at reviews, and that draws on that network to bring in those reviews, bring in those comments. The teaching, you use a network of tutors.
Absolutely.
That took something to actually set up and get running. So tell us more about that.
I mean, back to your point about networks, I think in the field of flow cytometry, the community is very open, very, very easy to work with, very, wants to share, which was different from some of the other societies I've been to. And so that really helped me build my network. I remember going to my first course, the course in Bowdoin, Maine, and everyone found out I was from Rochester. So they're asking, do you know Leon Wheelis? Do you know this person? I'm like, well, no. But I met Leon, after the fact. And it was just the community was so tight-knit, it really helped. And then within, building expert cytometry, already having a network of people like Alfonso, like Rui, in Europe, and having built connections overseas as well as in the US really helped pull those people together to help start the process. And then it just also keep me honest, I would have to say, about kind of the education, the training, making sure it's as high quality as it can be, and we're not things are being taking the best practices, and as things change, going back to talking about those. And so expert cytometry has really grown with the online presence, and also with face-to-face courses. And we've done some great ones with Alfonso in Europe, and ones in the US. I mean, we're working on organizing one in Poland, which is going to be very interesting if we can pull it off in hopefully when people can start to travel again. Not sure when that will be. I mean, especially since Europe is going to ban Americans from coming over to, rightly so. Don't get me wrong. I mean,
nothing to do with COVID. No,
no, not at all. Just
no, never, ever. Don't. But how do you juggle all of that with come on, you're running your own core facility. You are directing the gamut of core biological life sciences, and you have this business on the side as well. How?
Organization. It's really a matter of
Oh, good God. And you did the MBA as well. You did the other course as well. Jeez.
So the quick story about the MBA is my wife died 3 years ago from cancer, and trying to find something to fill some of that time. I had an offer to learn how to play the jazz saxophone or take the MBA. The guy with the jazz sax never called me back, so I never learned that. So I took the MBA, and at my stage in the career, being in core facilities for 20 years and having experience, it wasn't like I needed it, but I really learned a new way to think about things. And I think it's important for everybody to explore that thinking. But it becomes an organizational challenge to run multiple facilities. With the university, the first step was getting I'm a big fan of Jim Collins in one of his books, Good to Great. He talks about getting the right people on the bus. So I've got phenomenal people running each of the individual facilities. And so they don't need me to oversee them every minute of every day. And I set kind of the overall goal with them, and they run it. And that gives me time, especially at night, to work on other things.
That's great. I have to press pause a minute, guys. Just wait a minute. I think my neighbour's car's just been stolen off their drive. I'm not joking. Just bear with me. Actually, while I'm running off, Tim, can you? It's surreal. This is the most surreal moment I think I've ever encountered. Oh my! I've got to ring it in really quickly. Can you talk about your favourite publication, Tim? If you tell Alfonso about it, I will be back. I will just delegate this job.
Okay.
Talk about your favourite publication. I'll be back. Go for it.
Go for it.
That's your favorite publication is always tough. I think the one that I liked the most was this paper that I was a co-author on with this guy Jim Pallas's group, and it was called the Ontology of Erythroid Gene Expression, and they were trying to build an atlas of the different of the expression of different in different phases of the red blood cell. What was interesting about it that I liked is it was a challenge to identify and sort cells, because most of the stuff we do is lymphocytes, and they behave in a certain way. Red blood cells don't behave at all like that. We had to develop new sheath fluids. We had to optimize the catch conditions. We had to come up with ways to see red blood cells because there wasn't a lot to do. So we really, going through a lot of work to develop those processes. And it was kind of, really in the weeds, kind of fun, and it ended up in what I think was a great publication from, with the gene work that came out of that.
Okay, and Alfonso, what about yourself? What's your favorite publication?
While he was talking, I was thinking about that question. My favorite one, no doubt, is the calcium. The one we were talking about calcium flux in a continuous way. It's not because of the paper. I will not say the paper is a huge quality or anything like that. It's the history behind it. And it's because there's the acri on the back of that one. And that's my second funniest toy because of the history behind the acri. And it's when I arrived to Cytometry and I was talking with the crowd in acri that built up this little saying that was a flow cytometer. And you can close your eyes, push the button, and suddenly you run everything. And I couldn't believe it. And there was a beta test. They sent me the machine for beta testing. And during the first 3 months, I found out that application of being able to analyze and put things inside the tube and run them without stopping them and continues. So I asked for an extension of that time, and working with the Acri team at that time was fantastic because you ask a question and the engineer got it and they have implemented. And you ask a question to Leo or Kate and suddenly everyone was on board and your answer was just there. And suddenly on the following day, you have a software development. It was a fantastic period of time, especially because I learned a lot from the company, how the companies move, and working with that kind of people, I really enjoy. And of course, with the crowd here in UCD, preparing the samples and excitement We had an amazing sample that we were running and exploding all the glial cells, and I couldn't believe what was a drug. They made them exploding, and that was a real drug. Was ecstasy. They were given to the glial cells ecstasy, and the cells were like blasting. So all the history behind that paper is what makes that paper special.
I think that's a really good point. The fact that it isn't the highest impacting paper sometimes.
Mm-hmm.
It's just something special about a bit of work that, it's just that, and that's that contribution to it. I remember that work, and I remember you doing it at the time as well. Yeah. Remember phone calls with you about it and looking at some of the data early on and saying you're nuts. But anyway, before we finish off, I have a couple of questions. What are do you think the greatest unmet needs? Where do we need to see flow cytometry going in the future? I'm going to let one of you begin first for that one.
I think it's we're in a renaissance with new fluorochromes coming out, new ways to analyze data, new ways to trigger and explore cells, and I think better understanding of these automated techniques is important, because a lot of people use them, but they don't understand what they're getting out. Ryan Brinkman and folks like that are doing a really good job of trying to explain it, but the average person doesn't understand it. They just I want a Tisney plot of this or whatever. But really the development of new fluorochromes, and there's a big emphasis on things to be coupled to proteins, antibodies, but we're missing the time when molecular probes under Rich Hoagland was just developing fluorochromes and fluorescent compounds, and they didn't know what to do with them. But that's how we got calcium flux. That's how we've got reactive oxygen measurements, MBBR. We're not seeing a push to understand that internal development anymore. And I'd love to see some more probes being built up around other biological processes that we can refine.
Okay, and Alfonso. Tim's just giving you some really good buying time there, or just completely blowing you out the water with some really good words.
They're really, and Tim knows quite well this, and you as well, because you're always putting the flow on the top, right? On this scheme where you have genomics and proteomics and imaging and all the kind of imaging, and we can see all this imaging and the Rhapsody or the 10x genomics coming with single cell genomics and the spatial distribution that they have on the tissue, and you can see all this site of technology coming up with the Hyperion on the back, again with tissue distribution. I think this is what we are missing in flow cytometry, but on the other side is the speed, what we have in our favor, let's say that way. So we have imaging on the other side with imaging flow cytometry with the amnis. We are missing the sorting on that side. We are missing a lot of these, as Tim was saying, the possibility of analyzing all of these together in a quick way, in a visual way. So a lot of this is going into all these tools to visualize, to understand what is happening, to see that I have a dye and it's coming here, exactly where and how it's interacting. On the other side, all these interactions with the nanoparticles, with the EVs. The EVs is an area that is still growing up. A lot of technology for sure will be developed very soon in that direction, kind of flow cytometry side, with new tools, new markers to try to identify them with a smaller size micro fluidics, not nano or ultra nano fluidics coming up, to try to look at them, to sort them as well, to be able to do a good characterization of them, because at the moment we are limiting in 20. Who knows if there's something else even smaller than 20, sending things from one side to another, because we were talking about hormones a few years ago, and they are the principal messengers. Now we are talking about the EVs as one of the messengers or main messengers. So I think all of that interaction is where we are moving. And again, trying to put all this information together, a pipeline of all that information together. That's where the challenge is, all the artificial intelligence.
And I think from what both of you have said, the biggest challenge may actually be in the analysis of all this.
the amount of data that's being generated. I mean, Alfonso was saying it with all the imaging technologies coming out. When you can analyze on the Hyperion, 30 different, markers in a spatially defined manner. How do you put that together?
Yeah, we're fighting that at the moment with the NanoString DSP. That's quite a simple approach, but 40 colors, now going up into the thousands of RNA transcripts with spatial resolution. You still have to be asking the question and finding the answer rather than maybe trying to find what answers are in there. Yeah, we've got a long way to go, I think, which is why it's so exciting, isn't it? Huh?
Yeah.
Anyway, Tim, Alfonso, you've been great fun, really great fun. Thank you very much for joining me today.
Well, thank you. Appreciate
it's a good
it.
pleasure talking with both of you always.